Plant diseases, infections, and fungal pathogens are some of the most important subjects an arborist can understand because they directly affect tree health, structural stability, risk assessment, and long-term landscape management. Trees are constantly exposed to fungi, bacteria, viruses, nematodes, and environmental stress, but disease only develops when three factors come together: a susceptible host, a pathogen capable of causing harm, and environmental conditions favorable for infection. This relationship is known as the disease triangle. If any one of those factors is removed, disease development can often be slowed or prevented. Fungi are the most common cause of disease and decay in trees. Fungal spores spread through wind, rain, splash, contaminated tools, insects, soil movement, root contact, and infected nursery stock. However, many fungal pathogens primarily exploit stressed or weakened trees. Drought stress, overwatering, compacted soils, construction damage, improper pruning, poor planting depth, nutrient deficiencies, sun scald, and root disturbance all weaken a tree's natural defense systems and make infection more likely. Some pathogens are opportunistic and mainly attack stressed trees, while others are aggressive enough to infect relatively healthy hosts. Diseases like Dutch elm disease and sudden oak death can spread rapidly even in otherwise healthy trees under favorable conditions. One of the most important concepts in arboriculture is compartmentalization. Trees do not heal the way humans do. Instead, they isolate injury and infection through a process called compartmentalization of decay in trees. Trees create chemical and physical barriers that slow the spread of pathogens and decay. Some species compartmentalize very effectively, while others decay rapidly after injury. This is why proper pruning is critical. Flush cuts, topping, bark tearing, and unnecessary wounds create entry points for pathogens and overwhelm the tree's defense systems. Different pathogens attack different parts of the tree. Root diseases, such as armillaria root rot and Phytophthora, attack root systems and root collars, weakening both tree health and structural stability. Symptoms often include thinning canopies, chlorotic foliage, branch dieback, reduced growth, and epicormic sprouting. Mushrooms, fungal conchs, white mycelial fans, or black rhizomorphs near the base of the tree may indicate advanced infection. Canker diseases infect bark and cambium tissue, creating dead, sunken areas on stems and branches. Common symptoms include cracked bark, oozing sap, branch dieback, and dead cambium beneath the bark surface. If the canker expands around the trunk or branch, it can girdle vascular tissue and kill everything beyond the infection site. Wilt diseases invade vascular tissue and disrupt water transport. Pathogens like verticillium and Dutch elm disease fungi spread through the xylem, causing wilting, scorched foliage, chlorosis, and rapid canopy decline even when soil moisture is adequate. Some wilt diseases can spread through root grafts between neighboring trees. Foliar diseases affect leaves and needles and include anthracnose, powdery mildew, rusts, and leaf spot fungi. Symptoms may include spotting, curling, fungal coatings, chlorosis, and premature leaf drop. While these diseases are usually less structurally dangerous, repeated defoliation weakens trees over time by reducing photosynthesis. Wood decay fungi are especially important for arborists because they affect structural integrity and public safety. Decay fungi break down cellulose, lignin, or both within the wood. Serious structural degradation can occur long before external symptoms become visible. Arborists may identify decay through fungal conchs, mushrooms, cavities, cracks, dead bark, hollow sounds during sounding tests, or abnormal movement under load. Brown rot fungi consume cellulose and often cause rapid strength loss before major weight loss becomes obvious. White rot fungi degrade both cellulose and lignin, producing stringy or spongy wood. Once advanced structural decay occurs, no treatment can restore the original strength of the wood. Bacterial diseases are less common than fungal diseases, but still important. Fire blight causes blackened shoots that appear burned, while bacterial wetwood, or slime flux, produces dark oozing liquid caused by bacterial fermentation inside the trunk. One of the biggest challenges in arboriculture is distinguishing infectious diseases from abiotic stress. Soil compaction, herbicide injury, drought stress, overwatering, poor drainage, heat stress, and nutrient deficiencies can all mimic disease symptoms. Proper diagnosis requires evaluating the entire site, not just the visible symptoms. Arborists must also understand the difference between symptoms and signs. Symptoms are the tree's response to disease, such as wilting or chlorosis. Signs are physical evidence of the pathogen itself, such as fungal fruiting bodies, spores, mycelium, or bacterial ooze. Environmental conditions strongly influence disease development. Most fungal pathogens thrive in moist, humid environments with poor airflow. Wet weather, excessive irrigation, dense canopies, and compacted soils often create ideal conditions for infection. Prevention is usually more effective than treatment. Healthy trees naturally resist disease better through strong compartmentalization and vigorous growth. Proper species selection, correct planting depth, mulching, irrigation management, soil protection, and proper pruning all reduce disease susceptibility. Avoiding unnecessary wounds is especially important because injuries provide direct entry points for pathogens. Pruning is one of the most important disease management tools arborists use. Proper pruning improves airflow, removes infected tissue, and reduces pathogen spread. Pruning during dry weather helps reduce fungal spore transmission, and tools may need to be disinfected when working around vascular or bacterial diseases. Sanitation is also critical. Infected branches, leaves, wood debris, and stumps may harbor pathogens and should be removed when appropriate to reduce inoculum levels and future infections. Chemical treatments can sometimes help suppress diseases, but they are rarely complete cures. Most fungicides are preventative rather than curative, meaning they protect healthy tissues from infection, but often cannot eliminate pathogens already established inside the tree. Chemical treatment is rarely effective if the underlying environmental stress remains unresolved. Modern arboriculture relies heavily on integrated pest management, or IPM. IPM combines monitoring, accurate identification, cultural practices, environmental modification, sanitation, biological controls, and targeted chemical treatments when necessary. The goal is long-term disease management with minimal environmental impact. Not all fungi are harmful. Mycorrhizal fungi form beneficial relationships with tree roots and improve water and nutrient uptake, especially phosphorus absorption. Healthy mycorrhizal networks improve drought tolerance, root development, soil structure, and overall tree health. In California and throughout the western United States, arborists commonly encounter diseases such as sudden oak death, anthracnose, armillaria root rot, pine pitch canker, and Fusarium dieback associated with invasive shot hole borers. Understanding regional diseases is essential for proper tree management and risk assessment. In short, plant diseases develop when susceptible trees encounter pathogens under favorable environmental conditions. Fungi are the primary cause of most tree diseases, though bacteria, water molds, and environmental stress also contribute to tree decline. Healthy trees are better able to resist infections and compartmentalize damage, while stressed trees become far more vulnerable. Proper diagnosis, prevention, pruning, sanitation, environmental management, and integrated pest management form the foundation of effective disease control and modern arboriculture.